My name is Matt Duggan. I'm a senior biology major here at Wheaton College, and I've been conducting research with Dr. Nadine Morum of the biology department for the past almost 3 years now. We conduct research on an invasive hydroid species in Lake Michigan. This species originated in the Black and Caspian Seas and was carried to the lake, we think, in the ballast water of large ships. And because it's an invasive species, we hope to assess the predatory impact of this hydroid so we can see what kind of impact it's having on the native food webs in Lake Michigan. So in order to do that, we took two major approaches. One, we collected samples of Cordylophora at 8 harbors in the Chicago area and brought them back to the laboratory and dissected them to see what sort of prey items that they were ingesting and the different sizes of the prey items that they were placing. And then the second major type of experiment that we performed was to have live feeding experiments in the laboratory. And so we took colonies of Cordylophora, grew them in the laboratory, and gave them different types of prey to see how they would capture them and what sizes they could capture. And so by utilizing these two major avenues of investigation into the predatory impact of Cordylophora, we hope to see how exactly this organism will affect the native food webs in Lake Michigan. One of the things we found was that Cordylophora enjoys eating quite a lot of different types of prey items, including chironomids, which are a very important prey item for native larval fish. And so if Cordylophora is eating these chironomids in large numbers, that could be affecting competition with native fish and affecting possibly the populations of those native fish in Lake Michigan. And so we investigated the chironomid larvae ingestion in further detail in our live experiments and found further that Cordylophora is able to ingest very large chironomids, up to 2 or 3 times their size. But we also found that they're not ingesting them to large numbers, so that may not be having quite a large impact on the fish.
Are these the locations where you did your samples?
These are locations that we collected the Cordylophora. There are 8 different harbors highlighted here, right in the downtown Chicago area. We use scuba diving and scraping under the docks to collect the different samples. We actually collected the data over 2 different years, but we've been doing analysis and writing the manuscript over the past 3 years.
Okay.